to examine.
time we get started talking about the Calvin cycle, remember what happens in these cycles. The light reaction energy is captured, turned into either NADPH or ATP. That energy is sent to the Calvin cycle. In the Calvin cycle, carbon and oxygen from carbon dioxide will be reduced with hydrogen from a water molecule that was split in the light reaction until we came up with the basic sugar. This is the Calvin cycle. Notice one of the intermediates here is going to be glyceraldehyde 3. That was also found in the fifth step of the investment phase of glycolysis. They were taking glucose in glycolysis and breaking it down into a component. One of those is G3P. Now we are building it. Also remember there are three things that happen in carbon fixation, or the Calvin cycle. Carbon fixation is one, catalyzed by this enzyme Rubisco. It will be reduced with the electrons from hydrogen, and then it will regenerate the carbon dioxide acceptor, which is RUBP, which is ribulose bisphosphate, as we will see. On this particular setup right here, the steps that occur in through here, just know some things that you look at the top. It takes 3 cycles of carbon dioxide to wind up with the G3P. And that G3P is going to be used to make the glucose and the other things. Some G3P will stay in the system. Not all of it will come out to make the sugar, but some will stay in the system and regenerate that acceptor. Regeneration of the carbon dioxide acceptor, RUBP, that right there, ribulose bisphosphate. Then that will be in the system, Rubisco will come in, join up with that again, and continue. Brings in the carbon dioxide to go up. So, basically, the big thing to remember, carbon dioxide is coming into the system, being reduced to make a simple sugar. Eventually, remember that sugar is CH2O. This is carbon fixation cycle, but you see that happening. Unfortunately, getting carbon dioxide into the system, letting oxygen out is not as straightforward in every kind of plant. We have different kinds of plants that live in different environments. Some of those environments really suck the moisture out of anything. And if they're in a very, very dry, arid type of environment, whenever these plants open up their stomata to take in carbon dioxide, they're also letting out water vapors. And many of these live in areas where there is very little water. It's precious. They don't want to lose it. For that reason, there are some plants that have devised other ways in which to go through carbon fixation to get the carbon for carbon fixation. One of the things I do want you to know, photorespiration down at the bottom. And if we just read through this, it says on hot, dry days, plants close their stomata. That conserves the water. But because it's closed, it also will limit photosynthesis because there's no carbon dioxide coming. The closing of the stomata reduces the carbon dioxide and causes then oxygen to build up. These conditions favor an apparently wasteful process called photorespiration. We will see that plants are going to be divided into 3 basic groups of plants based upon how they do carbon fixation, how they get the carbon dioxide. Us, the plants that we have around here, if you notice our environment, we usually got plenty of water, and there's very few days where it's really so arid that plants are in danger of losing too much water. These plants are called C3 types. C referring to the carbon fixation. C3 types. The initial fixation of carbon dioxide via Rubisco forms the 3-carbon compound 3-phosphoglycerate. That's the cycle that we just looked at. That's pretty much our normal way. Plants here don't have to worry about opening their stomata. They can do it whenever it's necessary. In photorespiration, Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle. Produces a 2-carbon compound. Don't try to remember all of that. Just remember photorespiration is going to do things a little bit different. And notice photorespiration consumes oxygen and organic fuel and releases carbon dioxide without making any ATP or sugar. So what is the purpose of photorespiration? Basically, it's a way to get rid of the carbon dioxide and the oxygen that's in there, because it starts to basically get rid of the carbon dioxide or oxygen, because it's building up and the system doesn't need it. And there's no energy made. It's just a way of getting rid of the byproducts. Notice the middle. One of the purposes for this, anytime you start to get byproducts or products built up within a system, if they're not doing something useful, they're usually doing something negative. And in this case, photorespiration limits damaging products of like reactions that build up in the absence of the Calvin cycle. So since the Calvin cycle is not functioning, it doesn't want the parts of it in there, because they can do other things that would be damaging. Remember, things such as oxygens can become free radicals and damage tissues, the nucleic acid within the nucleus, whatever. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. Remember, going through the Calvin cycle and carbon fixation is just part. We have to have water. We have to have that hydrogen to make the sugar to do the reduction. And if we don't have everything that we need there, taking in that carbon dioxide and having that oxygen build up is not a good thing. So, that was C3 plants. Remember, basically, here's what I want you to remember. C3 plants can go through normal carbon fixation and the normal Calvin cycle. They have no limitations on when they can open up the stomata and take in carbon dioxide and getting oxygen. For plants who live in different conditions, they must change and do it differently. One of those plants groups go through a different processor called C4. There are different kinds of varied conditions. You can go out into the deserts and there is just very, very little water there at all. Now think about the air in those deserts, and if you've ever been out in any of the big deserts, it will literally, you won't ever sweat, ever, because it just sucks everything out into the air just immediately. But there is another condition that is as dry or drier, but there's a lot of water involved. Think about Hawaii. You've got many of those islands that are standing right here against the ocean, and the winds blow every bit of water away. Even though there's water in the air, and it doesn't draw it out of plants as much because there's moisture there, they still don't get much water. And one of the driest places, or it's supposed to be the driest place in the earth, is a little spot, I think it's in northern part of Africa, right on the ocean. But the winds just blow, they get something like less than 1/10 of an inch of rain every 10 years. And it's just sand. That's what it is. You don't find very much there at all. If they do, they're coming in and going out someplace else. So what I'm trying to say is things can be dry under different conditions. Now, C4 minimizes the cost of photorespiration by incorporating carbon dioxide into 4 carbon compounds. Remember, photorespiration tries to do 2 things. If you do have carbon dioxide in, then you don't have water that you can spare. You got to get rid of the carbon dioxide. Well, C4 plants do that a little bit different way. They have found a way to store that carbon dioxide by making other compounds out of it that won't be harmful. This step requires an enzyme, PEP carboxylase. PEP carboxylase has a higher affinity for carbon dioxide than Rubisco does. It can fix carbon dioxide even when carbon dioxide concentrations are very low. One of the big problems with photorespiration, those plants can't open up their stomata to get enough carbon dioxide in, but they do have some in there. They don't want that some. If they can't go through all the way the process to make the sugar, they want it gone. So that's what photorespiration does. It gets rid of the oxygen and carbon. PEP carboxylase is just a thing across evolution. It can take very small amounts of carbon dioxide and use that to fix the carbon fixation. I'm not going to ask you how all of that occurs or anything like with these bundle sheath cells. Just remember that PEP carboxylase in C4 plants can fix very small amounts of carbon dioxide, whereas Rubisco must have a higher concentration. That's in C3 plants. This is that C4 pathway. Once again, you don't have to remember that. The 4th type, these are desert plants, the succulents. This is called the Crassulucian acid metabolism, or CAM for short, CAM plants. Now, if you go out into many deserts, you'll find that the conditions change quite a bit once the sun goes down. During the daytime, there's an enormous amount of dehydration going on, but at night, things cool off and can get rather chilly. I mean, you can go out to the desert sometime and have it 100 degrees during the day and want to wear a jacket at night, a light jacket, because it cools off so much where it's at. Depending on the deserts, not all of them are the same, but it occurs. And so some of these desert plants at night can open their stomata. CAMs open their stomata at night, bringing in CO2 into organic acids. Most during the day, what they do is of the nighttime to build up a store, because they can't just keep it open all the time. They'll bring in carbon dioxide, put them into little organic acids they can keep on the side shelf, and then whenever they need it, release it from them. These shows here the C4 plants on islands, usually like Hawaii. You see sugar cane. They also, another example of this, are things like pineapples that grow. For the can. Sugar cane is an example. And even some types of the corn that we have that are resistant to drought and things like that have some of these characteristics. And the can are the succulents. And if you ever notice some of the big cactus that grows in the desert, things like that would be examples of these. So, thing to remember about this part is carbon fixation is taking carbon dioxide, reducing it with hydrogen from water that came out of electrolyte reaction, making a simple sugar out of it. Because plants live in different environments, they will have 3 different methods of incorporating carbon dioxide. Our type of plants, the most common type around the world, are C3. They can open stomata and take carbon dioxide in when they want. In C4, they can only take in very small amounts of carbon dioxide. So C4 plants use a different enzyme, the PEP carboxylase, that can go through carbon fixation with small amounts of carbon dioxide. And the fourth situation is CAM. CAM takes in carbon dioxide and stores it in organic acids until it needs it.
And what acids?
Organic acids.
Organic acids.
I was just thinking about that. I said it stores it in organic acid. Sounds like I'm saying inorganic. It's not. It stores it in acids that are organic. Okay? Yep.
So, excuse me. So we have carbon. So we have. You said 4 different conditions. I mean,
3.
4 different. Oh, 3. That's what I thought. Okay. So
C3, C4, CAM.
Thank you. Oh, and also for carbon fixation, so carbon dioxide is taken in and reduced,
Yes.
and then water is also used, and they make.
The hydrogen is used from water to reduce the carbon dioxide.
Okay.
Oxygen's given off.
Thank you.
Now, photosynthesis is fairly regular. I will give you some ideas to some of the questions that I will ask. Which are going to be what happens in the light reaction. What happens, water split, energy is absorbed, some ATP is made, NADPH is made. Photosystem 2 comes first. That's the photosystem where water is split, is in photosystem 2. Photosystem 2 and photosystem 1 photosystem 2 is primarily linear electron flow. In photosystem 1, it can be cyclic and generate some ATP. That is going to go down a second electron transport chain, starting with paradoxin, that is going to send the energy that was captured into the Calvin cycle for carbon fixation that we've just talked about. You should know the 2 electron acceptors, P680 and P700, what they do. But it really, there is not that much to remember. Starting here, we're going to go to this chapter on cell communication, then we get into how cells divide, genetics is coming up, chromosomes, heredity, things such as that. First part here, one of the most important things for any cell to be able to do, one of its most important jobs is communication. Cells such as your white blood cells have to be able to recognize, are the cells that's in your body yours? Are they bacteria? Are they foreign? That's the way your immune system works. Cells have to communicate in other ways. Think of your cells that need sugar in order to be able to metabolize and get energy. Well, it has to have insulin, which is produced in the pancreas, so there has to be communications between them. Your nervous system will communicate and tell cells to produce certain things, whether it's a hormone or a chemical that's needed. So communication is a big deal, very big deal. When cells injure or get injured, they have to communicate, and there's a variety of ways. They have to send some kind of signal. They have to give a message. And you can read through some of these things. There are some specific ones that I want you to remember, and I am not going to go through every single little thing in this chapter. I don't really think at this level we need to remember all of that, but make sure you listen so you can pick up what I'm going to be holding you responsible for. Notice that down at the bottom, the dilation of blood vessels is controlled by molecules, meaning so is the contraction. Your body needs to tell blood vessels when do we need more blood, when do we not. How does it do that? And that's going to be cell-to-cell communication. I want you to remember the definition for this: a signal transduction pathway. Where a cell can pick up a signal and make a response. In other words, a signal comes to the cell on its membrane, and it can turn that into a response. Here's a little example. Remember, we said cells on their membrane will have glycoproteins, which are carbohydrate parts to a protein molecule. They can be glycolipids, carbohydrates to fats. And the glycoproteins are the ones that makes up most of the signaling that cells pick up receptors. And keep in mind, now we often think of a signal as something that comes in a wave, like a radio wave we were talking about. It can also be a chemical. Up at the top, we see we've got cells with receptors. Notice there is one cell up here that is producing what is called the alpha factor that fits real nicely into the receptor on that A cell. Then the A cell can release a little molecule that fits real nicely with the alpha. So these are using chemicals or molecules that will fit a receptor whenever that receptor is released. And when that receptor fits, or the molecule fits into the receptor, it tells that cell, okay, now release A, or whatever it is that they release. That is in yeast. Now, what are they doing there? You have these cells that say that it is time to mate, so it releases a molecule and causes that yeast cells to multiply. Yeast cells come dormant whenever you see them in a pack. They actually have some live yeast in the middle of a big, if you've ever had your hands wet and got them into flour and see how that sticks to your hands, makes a coating, that's what those dead yeast cells do to the live ones. They actually make a coating around them, and then whenever they get water, they take in all that water, hydrate, and then the ones in the center that have been protected come alive. And these are some of the little messages that they can send, saying, we're here. And yeast actually go through two stages whenever they get hydrated. One is the hydration stage, which is a multiplication stage like this. Then they go into a metabolic stage in which they start to eat, where they tear down the glucose and start to make carbon dioxide released and make do what the yeast do. Local and long distance signaling. Uh, Not much for you to remember on that, just understand the fact that many of these cells, they can. And it's usually done by chemical messages. Chemical messages or messengers. There are some of these regulators I want you to remember, and it will start to make a little bit more sense to you. Notice here it says animal cells communicate normally with local regulators, cell to cell. Like one cell will release something that another cell close by will pick up and respond to. Long distance signaling is hormones. Hormones are used, they're still chemicals, and they still cause these cellular responses, but they just do it over a longer area, space. In animals, if you don't know, you will find we will have certain organs that will produce these hormones normally, such as the pituitary gland at the base of the brain, the adrenals that sit on top of the kidneys, the thyroid that controls your metabolism, so forth. So these are usually organs that make these hormones. Epinephrine is one that your body actually produces. We call it norepinephrine or noradrenaline and things, but it works like adrenaline does, but it's a synthetic. It's used a lot in anesthetics. Everybody's heard of EpiPens. Epinephrine is a drug that if you put it into, for example, your trachea or respiratory type of epithelial cell, it will cause them to dilate. However, if you put it into the blood vessels, it will cause them to constrict. It does exactly the opposite based upon what tissue. What do you think would be the differences? Type of receptors. No, the drug's the same, but it will cause two different responses, and it's based upon what type of receptor is it going to. EpiPens are designed so that you can, if you get stung by an insect or have an allergy, it will go in your system and it will go all the way through your system, and whenever it gets into the smooth muscle of the tracheus, it will cause that smooth muscle to dilate. At the same time, it's causing the smooth muscle of the vessels that are pushing it to constrict, which is not a bad thing. What happens when the blood vessels constrict? Blood pressure goes up, so it's got more force to move that epinephrine to the tissues needed. So your receptors are a key here. These, I want you to remember, but they are very simple. Just remember, a cell picks up the reception, it will send it to where it needs to go to within the cell, and then it will respond. So reception, transduction, and response. Here is an example. You've got a signaling molecule, here's a cell with a receptor. Whenever that receptor is stimulated by that molecule, it will start to release molecules down that transduction pathway, and then it will give a cellular response, whatever that response is. You will hear this term a lot, and I do want you to remember it, ligand. A ligand is a molecule that causes a signal to occur. If causes a signal. And I want you to remember that most of these signal receptors are in the plasma membrane of the cell and are these proteins. I am not going to make you remember these three types of proteins here. When you take cell biology and get into it a little bit deeper, you will go over those then. So I'm not going to make you remember those now. G proteins, though, are very common, and they are a very common way that all of this works. Once again, the signaling part, the receptor site, as you see up here, is going to be hydrophilic. It's going to be where water occurs. These coils that go through the membrane, the liquid tails, are going to be hydrophobic. And then the part where the reception or the transduction occurs is going to be also hydrophobic. So this is what it's showing. The big thing I want you guys to remember from this is not what each of these sectors are. No, a ligand causes a signal, starts a signal. It's the molecule that starts the signal. And I am not going to spend a lot of time on any of this other stuff because in cell biology, you're going to be going way back over this just as deep or deeper again, and I don't see the point of doing it twice right now. So I'm not going to talk a lot about the receptors, but I'll tell you what you need to remember from them, which is what we're just skating over this right now. And because that is something that is hit so heavy in cell biology, which I also teach. But I want you to have an idea of what's happening, not necessarily how it's happening right now. All of these terms, the protein kinases and phosphatases, that gets very much complicated. We'll get into that cell. Now, here's where we're at. Cells on the membrane usually will be a protein that will act as a receptor. When that receptor is stimulated by a ligand, that molecule that will fit to the receptor and cause the signal to start, that's the reception part. Then that protein will send a message to whatever part down in through the cell to get things started. That's the transduction. And then whatever that signal is, where it's gated to or where it's targeted to, will cause a reaction. Maybe it's release of another molecule, whatever it is, but it will cause that to occur. Now, we're looking at one other thing I want you to remember right here, second messengers. A second messenger, as it says, non-protein, water-soluble molecules that spread by diffusion in the cell. Now, what in English that means, we get the ligands that attach to the receptor, causes the transduction, the signal to be sent, and then a reaction. Part of that is can be the cause for the release of second messengers that will go inside the cell now and do something. So the protein on the surface has been stimulated and says release these little guys, so they're released and they go through the cell. One of these I want you to remember is at the bottom, these two, cyclic AMP. What does that sound like? you had to guess what is that, what is it? Adenosine monophosphate. We have looked at ADP, adenosine diphosphate, triphosphate, the energy carrier. This is the form with only adenosine and only one phosphate. And calcium ions. Where is calcium important? You'll find in muscle cells in reactions and having muscles to contract. Cyclic AMP is a very important one of these. Notice here, adenyl cyclase is an enzyme that converts ATP to AMP in response to that signal. You don't have to really understand the process. Calcium, remember, is the other second messenger to remember. Some of these others I'm not going to remember. I'm not going to spend that much time on because we got some other stuff that won't be covered in cell biology we got to get to here. So, you're not going to miss out on anything, I promise you. I just want you to have a basic understanding of how these things actually do occur. One other term I want you to notice is up here, apoptosis. Programmed cell death. So when the cell starts to get ragged around the edges, so to speak, and it's not functioning 100% the way it's supposed to, it will die. It will die in such a way that other things will come in and kind of isolate that little cell, so all of the junk, like the enzymes that can be harmful inside, don't get out into the tissue and hurt other good cells. It's just a neat way of getting rid of the cell and all of its harmful parts inside. Notice it says apoptosis prevents enzymes from leaking out of a dying cell and damaging neighboring cells. So here's what I want you guys to remember from this. Cells have receptors that are mainly proteins. That receptor can pick up a molecule that will stimulate the cell to have a response of some kind. Sometimes what it will do will be the cause to release second messengers inside of the cells, such as calcium, cyclic AMP, and you'll find, if you ever study anatomy, that this calcium is a huge, big deal in muscles, contractions, and so forth, the cisterna and things on them. But they will cause a second release. That is the way cells communicate. Now, when you get into cell biology, you will then see all of these chief proteins and how that actually does occur. But for now, that's what I want you to know. I want you to know that cells will protect the surrounding tissue by a process called apoptosis, so that whenever they get ready to die, they will do so by being walled off and in a clean environment. Photosynthesis and the cell cycle will be the key two parts on this next exam that comes up. I would predict we will get done with the sales cycle. It's not that long. We will probably get done, we've got next Wednesday. Then we have the Monday of Thanksgiving, so the Wednesday of Thanksgiving week is when you attend your next exam, before Thanksgiving. That way you won't have it lingering over top of you. And the school does not give us permission to give exams early so people can leave. We get in big trouble, we get warned over that, people want to leave, go on vacation, they say no. So you're gonna have to have some special permission from somebody besides me. Now, we still have one person that's gonna be taking the exam, but I'm gonna give these back to you, but I'm not gonna go over these. So if you wanna ask me a question about it, we'll go over them next time. What I'm going to do, what I'm going to do is call your name out, you can come and get your exam.